In this video, we will discuss about different strategies of the bacterial engineering to target tumors. In the previous video, we have discussed about how this bacteria can be used as a drug delivery tools, and here we will discuss about different strategies by which this bacteria can be engineered to target to the tumor sites. So, first engineered approach that is discussed in the panel A is about doxycycline induced gene expression system. In this case, the gene system within the bacteria has been modified in a way that it will be regulated by doxycycline. For examples, when there is no doxycycline, this TETR will be produced through this TETR gene, and that will inhibit the expressions of the tumor targeting genes, which are controlled by these two promoters, and these two promoters are being inhibited by these TETR repressors. However, when the doxycycline is induced, the doxycycline will react with these TET repressors, and thereby they will not come and interrupt these promoters, and thereby tumor targeting genes can be expressed when the doxycyclines are present within the system. So in this way, the two different kinds of tumor targeting genes can be targeted. So other approach is about the systematic thermal switch system. So this is based on the thermal or temperature based activations. So when in the physiological temperature, what will happen is that this TCL42 is not TCL42 will prevent the expressions of this BXB1, and because of that, the tumor targeting genes are not being expressed. So what will happen when in order to activate this system, we need to somehow deactivate this TCL42. So this can be done by focused ultrasound at the site of tumors, and by which at around 42 degree temperature, this TCL42 will stops or prevents the translation of this TCL42, and thereby this will cause the burst of BXB1 gene in the tumors or in the tumor microenvironment. And this BXB1 genes, what it will does is it will reorient this attP B and attP genes. We can see that in the case when physiological temperature is there, they are in the different orientations, but when it is activated using the ultrasound at 42 degree, the orientation change, and this will cause the translation of these P7 promoters. And here the tumor targeting genes have been designed to be controlled by P7 promoters. So once the P7 promoter has been translated, then the tumor targeting genes will also be transduced, and thereby they can act upon the tumors, as at that time these things are being activated once the bacteria reach the tumor sites, so that the non-specific effects are also lower. And next one is about the surface engineering of the bacteria. So in this case, the two different approaches are been tested. First, by engineering the bacterial surface to target AntiCD20 on this VHH protein, and at the same time they also engineered the bacterial surface with RGD domain and the same OMPA protein. So in this way, this will increase the specificity of this bacteria towards the cancer cells. At the same time, what will happen is that they will have these drug-loaded nanoparticles. These drug-loaded nanoparticles are coated on the bacterial cell surface, and these drug-loaded nanoparticles, when they are in the host system or in the in vitro conditions, in the in vitro conditions, these drug-loaded nanoparticles are then coated to the bacterial cell surface, and thereby when they reach to the site of the tumors, this drug-loaded nanoparticles can act upon the antigens against which these nanoparticles are being designed, and these are designed against the antigens that are specific to the tumors. The two examples here shown is against the cancer cell expressing CD20 or cancer cell expressing integrin alpha V beta 3. The last method is this macro nanobot, where bacteria can be used as a nanobot, for example, and this nanobot can be activated with heat using the alternating magnetic field. Using the alternating magnetic field, the nanobot will be activated, and this activations is also been coupled with this fluorophore release, and thereafter this heat will cause the translation of this plasmid that release bacteria lysate protein. At the same time, another plasmid will secrete this anti-CD47 antibodies, and these two combinations will help in targeting the tumors that has the first this what will happen, this nanobot, because of the alternating magnetic field, will cause the activation of the bacteria that will release this. It will cause the activation of the plasmid that will release this bacterial lysate protein. This bacterial lysate protein will lysate the bacteria, and thereby it will cause the release of the anti-CD47. The anti-CD47 can target the tumors that has the CD47 expressions. And one of the other examples that is shown here is that the bacteria has been engineered to target this hepran sulfate proteoglycan, and these are also found on the cancer cells such as colon cancers. So these modifications can be used against the colon cancers to improve its therapeutic efficiencies. So these are some of the approaches that are mentioned in this figure. If you need to understand more about their bacterial engineering, tumor target methods or approaches, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.